Okay, so there are different types of electrical signals. We've got graded potentials, we've got action potentials. And these are basically different amounts of the same type of event, right? Graded potentials are going to be small, sort of inconsequential, trivial changes of the membrane potential. These communicate over short distances of time. So they're not as meaningful, they're not as sustainable as action potentials, but they are changes in the membrane potential. So in this part of this schematic here, we can see the small change in the membrane potential. These are very tiny, inconsequential, not sustainable changes of the potential. Action potentials, on the other hand, are much larger. They're going to communicate over longer distances, and they're going to really send signals from one cell to the other. Whereas graded potentials are going to maintain or stay within a single cell or a single part of the cell. And we can see on the right here that the action potential is a change beyond -55, right? And it's a sustainable, meaningful, positive change in the membrane potential that has the ability to do work. Okay, so let's describe a few parts of the neuron, just so that we have some context, and speak about these graded potentials a little. So speaking about different types of potentials here, and let's start by looking at the structure of a neuron. Okay. Okay, so we start off by looking at the important parts, the soma, or the cell body, which is this part of the neuron. This is what's receiving most of those electrical signals. We've got the dendrites, which are these axonic projections. We've got the synapse. The synapse represents the communication between one neuron and another neuron. We'll speak a lot about the synapse in a future packet. We've got the axon, which is this length of the neuron, that communicating bit of the neuron. This is called the axon. We've got this really important region here at the neck, so to speak, of the neuron. This is called the axon hillock. This is a really important region for determining whether or not an action potential is fired. We're going to speak a lot about that region. We also have the terminal bouton, or the axon terminal. This is the distal part of the neuron, so signals are moving from this direction towards the axon terminal. And then the axon terminal will eventually communicate to another neuron, as we can see here. All right, now let's divide or distinguish what parts of the neuron we have different potentials. So we talked about graded potentials being those small changes of the membrane potential. Those are usually limited to the dendrites and the soma or cell body. They don't usually travel beyond the axon hillock. Whereas action potentials are going to be established at the axon hillock and travel all the way down to the axon terminal. Okay. Now, we can also describe these two neurons in terms of their relationship here. So because this is the synapse, we can describe this neuron as being the presynaptic neuron. Presynaptic meaning before the synapse. And then this neuron that it's communicating to being the postsynaptic neuron. Okay, that's going to become more relevant as we speak about different types of synapses. Okay, so this is basically the structure of how neurons communicate and different features. And we'll come back to this drawing and point out where some of the channels are. But for right now, we're really concerned about distinguishing where graded potentials occur and then where action potentials occur. Okay. Okay, so graded potentials, we said, are initiated by a stimulus. They are small changes of the membrane potential. Their magnitude can vary, so they can be small, they can be medium, they can be large. We'll see that that cannot happen with action potentials. They're spread by what's called electrotonic conduction, which basically describes that separation of charge, the inside, the intracellular fluid of a neuron being negatively charged, the extracellular fluid being positively charged. And just in a similar way that you have a battery with that separation of negative and positive having a potential to transfer a current or a force, we have that same type of conduction happening here. These are decremental, meaning that they will decay or decrease in their magnitude as they move away from the stimulus. So at the site of the stimulus, which is here on the cell body, we have strong graded potentials, but as they move towards the axon hillock, they eventually dissipate or decay, and that's what we mean by decremental. Now, to understand why they sort of decay or dissipate, think about water moving in a hose. So water moving in a hose that has holes in it will eventually lose the pressure and the quantity of that water as it goes from the faucet end down to the distal end of the hose. And a very similar phenomenon happens here with the neuron. So as we move away from the source of the stimulus, we lose that potential energy in a similar way that water is leaking through a hose with holes in it. So in this image, we can see that there's a small potential here, there's a stronger potential here, but neither of these have enough of a change to generate an action potential. Okay. And at the bottom here, we can see how that potential dissipates from the site of the stimulus as it moves towards the axon hillock. Now, the purpose of graded potentials is to determine whether or not an action potential will occur. So at threshold, and the threshold value is negative 55 millivolts, we then have enough of a meaningful change to stimulate an action potential. But until we get to threshold, all we have are those small changes, either depolarizing or hyperpolarizing the membrane potential. So threshold is described as the value or the membrane potential at which we can then trigger an action potential, and that's about negative 55 from negative 70 from our resting point. And then we have different directions of changes of graded potentials. Excitatory changes depolarize the membrane, such as this, making it more positive or less negative. Hyperpolarizing or inhibitory changes make the membrane potential a little bit more negative or less positive, and that can be this one on the right, on the left. Now, these small changes have the ability to sum. This is called summation, and summation is basically clustering these graded potentials together and then determining if there's enough of a change to fire an action potential. So we have two general ways to sum graded potentials. There's temporal summation. This is basically summing in time. So at the top here, we can see that if we have one graded potential and another graded potential fired at the same spot on the neuron, if they're close enough in time, we can simply add them together, and then we may be able to get to threshold and fire an action potential. So the same stimulus or the same spot on the neuron, and if they're close enough in time, we can sum them together. Spatial summation happens if there are stimuli happening at different places on the neuron. So different spots, but they're happening at the exact same time. And we can also sum those in space, right? So we have one graded potential happening at neuron Y, the synapse between neuron W, and then another graded potential happening at the synapse between neuron Y. And if we add those two stimuli together, those two graded potentials, we may or may not be able to get to an action potential. In this case, we didn't because one was a hyperpolarizing event and one was a depolarizing event. But if they were two depolarizing events, we would be able to get to negative 55, okay? So these are just two mechanisms of clustering those stimuli and then determining if we have a strong enough change to get to that threshold and then fire an